Okay, so as we move into this next section of our video lectures, we're actually going to have two videos that go with this section. I'm going to finish up. We had a little bit from cells that we didn't get to that we need to talk about. So the first video is going to be just getting us through mitosis, talking about cell division, and then we will pick up with the integumentary packet. So two videos to watch this time. Before I get started in the video lecture, I wanted to remind everybody that our next face-to-face meeting, which will be October 12th, we are going to start class in the auditorium. So at 4 p.m., you will report to the auditorium over on the hospital side of things. And then we will be there from approximately 4 to 5. At 5 o'clock or somewhere around there, we will move over to the classrooms where we have been, and we will then carry out lab. So we'll still be in those two classrooms when we get back over to the other building, but just so that we can get through lecture as a large group and have the space that we need to in order to be able to do that. So October 12th, starting over at the auditorium, move over to the classrooms when we're ready to do the lab side of things. And I will keep posting that and keep reminding people just to make sure we're all on the same page. And since that hasn't been the norm up to this point, that nobody forgets and goes to the wrong place for that class. So in starting with the information that we need to go back and cover, we need to talk about cell division. So this is still in that cells packet. We have moved through a majority of the packet, but cell division we didn't have time to get to, and I do want to talk on that. So when we talk about cell division, there are two different ways that cells divide, and we'll talk about mitosis now as we are covering the cells, and we'll actually pick up the other type of cell division when we get into our reproductive chapter. Now, we have two different types of cell division because they accomplish two very different things. So when we look at cell division using the process of mitosis, the goal of mitosis is to either replace cells that have died, to reduce the size of the cell because it's getting too large and the nucleus can't take care of everything. Um, Remember, the nucleus is the brain of the cell, and so it has to monitor everything that is going on in the cell. As the cell size increases, the nucleus becomes overloaded, has too much to monitor, and it can't take care of everything any longer. And eventually, things don't happen the way they should, and the cell dies. So, cell division is a way to get that large cell reduced down to two smaller cells so that the nuclei can manage everything that's going on. Think about it like a classroom. If you have a kindergarten teacher who has 10 students in her room, she can monitor the students, watch what they're doing, and make sure that everybody's on track with what they're supposed to be doing that day. If you give that same kindergarten teacher 300 kindergartners to watch all at once, now that becomes a little bit more difficult task and something's going to get away from her. So same concept when we move to the cells. As the cell gets too big, nucleus can't watch everything, and something's going to break down, and the cell will ultimately die. So cell division, mitosis, happens to replace cells that have died or become damaged. It will help the cells reduce their overall size if they're getting too big. And the third reason that we have the process of mitosis is for growth and development. You guys are not the same size that you were when you were 5 years old. You're not the same size that you were at birth and at fertilization. So there is a process of needing more cells to be produced in order to allow for that growth and development to take place. So three reasons that we have mitosis. When we get into meiosis, which is the other type of cell division, we'll cover the reasons for that. Now, the part of or the process of mitosis is going to involve different phases. So there are the first phase of mitosis is called prophase. And if you break down the word prophase, pro indicates first. And so this is the very first part of mitosis, which is the actual process of the cell dividing. Now, some of the main things that are going to happen in prophase. Number one, the cell is going to do a lot of the things that it takes in order to really get ready for the process of cell division. So we have the chromatin, which is your DNA, the information for how to make you. All of that genetic information is held within the nucleus. Now, when the cell is not dividing, your DNA is allowed to be very relaxed. It spreads out and it really has no shape or no form to it. If you would imagine taking a string and holding it up over a table and then dropping it, and how that string would then just kind of lay on the table after you dropped it, that is the same formation of your chromatin. There is no exact shape, design to that genetic information at that point. It's just very relaxed, spread out, might kind of have that spindly appearance, like I said, like that string laying on a tabletop. Now, as the cell starts to move deeper into prophase, that chromatin needs to start to take some sort of form. And typically, when we talk about DNA and the true form of it, it's going to take on an X form. So if you've seen a teacher or an instructor at some point draw a chromosome, they will use that X shape as its form, and that's what it typically looks like, is somewhat like an X. So that tells you that that DNA, that genetic information is starting to pull together. It's starting to migrate into its identifiable form, and it looks more like an X. So we start to see that relaxed DNA start to group together. The other thing that we see in prophase is that those centrioles, now we talked about them briefly in class. The centrioles are two structures that spend a majority of their time floating around the nucleus. And when the cell gets ready to divide, one of the centrioles goes to, if you think of a cell kind of being like the Earth having a north and a south pole, one of those centrioles will migrate to the North Pole, the other centriole will migrate to the South Pole. Now, once they get there, they will take on a little bit more purpose as we get deeper into the process of mitosis. So for right now, they're just kind of taking up their or getting to their places for what they're going to do later on. The other big thing that happens in prophase is that the nuclear envelope is going to dissolve. So that layer that is containing and holding all of the material inside the nucleus is going to break down. Now, this is huge because it's that nuclear envelope, that membrane, that is holding your DNA in the nucleus and making sure that it can't go out and about. So when that nuclear membrane breaks down, now the DNA is able to migrate and move through the cell. And the only time it is able to do this is during mitosis. Any other time, that DNA is kept within the nucleus to make sure that it is well protected and cannot become easily damaged through things that are going on in the cell. Nuclear membrane breaks down. Now the DNA can go where it needs to in the cell, and that will play more of a part as we get deeper into the process of mitosis. The other thing that we have by the end of prophase is all of the organelles and your genetic information has a duplicate copy. Remember, the whole process of mitosis is to go from this one cell that we have into two new cells. And so as this happens, we need to make sure that each of the cells is going to have a full copy of everything. So all of the organelles are going to be duplicated. So when that one cell divides, each cell has its own set of organelles, and it also has a copy of the DNA or your genetic information. So a lot goes on in prophase. That's one of the reasons why it is the longest phase. Now, you can't ever put a time on mitosis. You can't say, well, prophase takes an hour or it takes a day and a half. It happens at the rate that it needs to happen, and it will vary from one cell type to the next. So there is no specific amount of time, but when you compare it to the other phases of mitosis, that is where the cell is going to spend a majority of its time. Now, when we get into metaphase, uh, metaphase is known as the middle phase, and one of the reasons for that, meta, the prefix indicates middle. And so we use that term, and we're really referencing what is happening with the chromosomes. So now the chromosomes look like X's. And in the human, we would have 46 total chromosomes. Now, some people will talk about chromosomes and they'll say 23 pairs. And the reason that we can reference them as either pairs or just talking about all 46 is because each one of your chromosomes has a twin. So there's another chromosome that holds the same information, and that is why that 23 pair reference, like I said, can be used. What happens during metaphase is these chromosomes will line up in the middle of the cell, and those two centrioles that we talked about earlier, Now they start to move into action because they're at the north pole of the cell and they're at the south pole of the cell, and they start producing this protein thread called spindle fiber. Now, what happens is that spindle fiber starts to extend out of those centrioles and starts to make its way towards the middle of the cell where all of those chromosomes are lined up. So we're going to go back to that Earth reference. If you think of the cell being the Earth, we have the North Pole, we have the South Pole, and then right in the middle, we would have the equator. So at the North Pole and the South Pole, we have a centriole. And right on the equator, we have these chromosomes all lined up straight across. Now these centrioles start producing the spindle fiber, and that spindle fiber is going to migrate towards the equator. And once it gets to the equator, it's actually going to hook and latch onto those chromosomes. And then it starts a tug of war. So you have spindle fiber coming from the North Pole, latches onto the chromosomes. You have spindle fiber coming from the South Pole, and it latches onto the chromosomes. Once they latch onto the chromosomes, the process of tug of war starts. And the spindle fiber from the north side will start pulling those chromosomes towards the north side. And the spindle fibers from the south side start to pull the chromosomes toward the south. And the chromosomes actually get ripped in half. So now those chromosomes, as they get ripped in half of them will go towards the north, half of them will go to the south. And this ensures that when the cell actually divides and goes from being one cell to two, that each of those new cells is going to have a full set of your genetic information. Now, one thing that I need to point out very quickly is that when we talk about your chromosomes lining up in the middle of the cell, it isn't somewhere around the middle or pretty close to the middle. This has to be precise. Spot on accuracy is of the most importance, that those chromosomes are dead center, middle of the cell. And the reason that is so important is because as that spindle fiber migrates towards the chromosomes to begin that tug of war process, as soon as the spindle latches onto a chromosome, it starts to drag it back to its pole. So if the spindle fiber from the north side would reach those chromosomes first, because the chromosomes were not lined up in the middle, maybe they're just a touch closer to the north side, that spindle fiber would get to those chromosomes first and it would drag those chromosomes to the north. South would never have latched on, so the chromosome would not get ripped in half. All of that genetic information would go to the north side of the cell, and that means the south cell didn't get any genetic information. That cell would have no chance of surviving. So those chromosomes lining up in the middle is huge, so that those spindle fibers hit the chromosomes at the exact same time so that they can do that tug of war process, ripping the chromosomes in half so that that genetic information moves to the ends of the cell. So when the cell actually divides and cuts into two, those new cells have all the information they need. The next slide talks about anaphase, telophase, and cytokinesis. Now, anaphase is what we call the movement phase, because now those chromosomes have been ripped in half, and the actual process of cell division happens through the equator. So everything clears out from the middle of your cell. All the chromosomes migrate to the north or the south pole. All the organelles will migrate to the north and the south pole, making sure that both ends of the cell have a full copy of everything that they need. Then you're going to start to see the cell really prepare for the next phase of telophase, where the cell you'll start to see the physical process of the cell dividing. So anaphase is the movement phase. Everything is on the move to make sure it gets to either the North Pole or the South Pole and clears out the middle of the cell. Now in telophase, what happens is we typically think of a cell being nice and round, but at the equator on both sides, the cell will start to pinch in, and you'll actually start to see these indentions on the side of the cell, and it no longer it looks like a circle, it starts to look like a peanut. So you have this round top, and then it indents in the middle, and then you have this round bottom. And so it takes on that peanut shape, and the cell will continue to indent on the sides until it cuts all the way through the middle. The cell is allowed to separate, and then that process is known as cytokinesis. So cytokinesis actually means division of the cytoplasm, and that is where you would go from your one cell that you started the process with to 2 new cells. So that indentation that you get on either side of the cell, I also need to mention, is referred to as cleavage furrow. So the indents on both sides of the cell that you would have, kind of making it look like that peanut or that hourglass, that is your cleavage furrow, and it just keeps pushing in, cutting through the middle until the two cells, or until the cell is separated into 2 new cells. Now, this process of mitosis is very regulated. Like I said, we don't have exact times. You can't say this one takes this, X amount of minutes, but it is something that is very regulated. And you have a special protein in your cells. If you're looking at your cancer slide, you have a special protein in your cells that is known as cyclin. And cyclin is really what is in charge of making sure that your cells only divide when you need them to. So cells are only going through the process of mitosis when it is too big and it needs to be reduced down in size, when you're growing and you need more cells to accommodate that growth, or when cells have died off and you need new cells to replace them. That is the only time that your cells should be going through the process of mitosis, and it is cyclin's job to make sure that that process is regulated, making sure that it happens the way that it should. In the case of cancer, something breaks down with that cyclin protein, and instead of regulating it to make sure that you only get new cells when you need them, it breaks down and now cells are being produced and you don't need them. So you get a mass of cells known as a tumor that you have no need for in your body. The problem with those tumor cells is they're going to now need an energy supply. They're going to need blood vessels serving them. And depending on how those cells grow, if they start to take over the surrounding cells around them or make it very difficult for the surrounding cells to do their job, then you obviously are going to see a breakdown in your overall health. So when we talk about cancer, we're talking about malignant, in which the cells are aggressive and taking over the structures and tissues around them, or it could be benign. You have a mass of cells, but it's staying contained to itself. It's not impeding or doing anything to the tissues around it. The last one talks about cell death, and cell death is what we call apoptosis. It is cells that are from the get-go programmed to break down. They have a certain lifespan, and once they have served their purpose, they are supposed to get the self-destruct message and just kind of break down, and you wouldn't see them or notice them anymore. Some of the biggest places where we would use this cell death process during embryological development, you actually have what looks like a mitten over your hands and your feet. So as the very small intricate parts of your fingers and your toes are being developed, that mitten is going to help hold everything where it needs to be to make sure that that development is allowed to take place. Now, once that's all developed and it doesn't need that extra protection kind of holding everything together, that web is supposed to go through the process of cell death, and those cells of the web are supposed to break down and dissolve away. Sometimes those cells don't get the message and they don't go through the process of cell death. So when you hear of people that are born with web toes or web fingers where the skin between the fingers is still there or the skin between the toes is still there, for whatever reason in that particular individual, those cells did not go through the process of cell death. They are still there at the time of birth. And depending on how severe is not the word that I want to use, but how much of it is left behind will determine what they have to do in order to fix it if they are able to. The other thing, it would be under the tongue. If you've ever looked under your tongue, you'll notice that there is a very thin piece of tissue that holds the tongue to the base of the mouth. That actually during embryological development goes all the way out to the tip of your tongue. So that skin extends all the way out, which would make it very difficult, if not impossible, for you to lick your lips, for you to stick your tongue out, do a lot of the things, move your tongue in a lot of the ways that we do for letter formation, sound formation, things like that. So again, supposed to dissolve and go through the process of cell death. Those cells don't always do what they're supposed to, and you do have babies that are born and that skin is all the way out to the tip of their tongue. Usually they can go in and clip it away, and it's not that big of a problem. But we do have places in our bodies where we have extra cells at some point in time that are there for a purpose. Once they've served their purpose, they should die, and we wouldn't see them or need them again for the rest of our life. So that kind of takes us through the process of cell division and why the process of cell division is so important. Making sure that all the parts of the cell get delivered to where they are supposed to be, so that when the cell actually divides, each of the new cells that would result from the process of mitosis has a full copy of your DNA, has all of the organelles that it would need in order to survive, and that everything is aligned and organized in the proper fashion. As soon as your cells fully go through mitosis and cytokinesis has been finished, the nuclear membrane would reform, containing all of the DNA and holding it again in that nucleus until the next time that the cell would need to divide, so that it's, again, getting that level of protection that it needs. So, very quick rundown on mitosis and the process of cell division. Like I said, when we get into reproduction, we will talk about the other type of cell division, and you'll see why it just is better to hold off on that discussion until we actually get into the reproductive chapter.